Thank you. A very exciting talk. I think has a very great summaries about targeting KRAS mutation. And so today, I think I really have the honor to work with Dr. Zha, and thank you for the organizing committee to be here. And I'd like to introduce Dr. Sher, and probably you know her better than I do, right? And had a very impressive CV. I think she received all her education from National Taiwan University and really built up her career at the China Medical University, and starting from the postdoc position all the way rising to full professor and also the director of Institute of Biochemistry and Molecular Biology. And so I think I already asked the question, what's the alternative? Maybe just not only targeting RAS, right? And what about other alternative and targeting for pancreatic cancer? So without further ado, Professor Sher, please. Thank you.
Okay. Okay, thanks for the kind introduction. I'm Yuh-Pyng Sher from China Medical University. It's my great honor to present our study about the how KRAS degradation. So my title is ADAM9 inhibition promotes KRAS degradation in pancreatic cancer treatment. And basically, we find a novel mechanism about KRAS degradation by targeting ADAM9. And based on it, we developed a small compound to promote the KRAS degradation in pancreatic cancer treatment. And it not only promotes the KRAS degradation, but also enhance the chemotherapy treatment. Okay, according to the Director Sher's introduction description, we know KRAS is the major target for pancreatic cancer because 90% pancreatic cancer with KRAS mutation. The current FDA-approved drug is specific target to G12C. However, G12C mutant only 1% in pancreatic cancer. So the FDA-approved drug may have limited treatment in pancreatic cancer. There's a lot of hurdles in KRAS inhibitor development. One is there are 12 mutation types in KRAS mutation, and also multiple KRAS mutant in one pancreatic cancer tumors. That means only target one KRAS mutant may not be enough to treat pancreatic cancer. So we need a broader strategy to target most KRAS mutant types. So that's why Revolution Medicine, he developed a molecular glue to block the RAS G12 mutant. So can we find an alternative strategy to treat to target all types of KRAS? Yeah, KRAS signaling is tightly regulated. It needs the outside signal of like EGFR signaling to make a KRAS on and to conduct downstream signaling. And when it's active, it also produces some inflammatory mediator, and that can activate some receptor tyrosine kinase to amplify the RAS signaling. So how to target pan-RAS mutant? Our strategy is to interrupt the KRAS amplification loop to finding some important KRAS mediator. So in our study, you find the ADAM9 is the important RAS modulator. So what is ADAM9? ADAM9 is the disintegrin and the metalloprotease 9. It is located on the cell surface. It is overexpression in most cancer types, and its protease activity can shed in some substrate to enhance the cancer malignancy. For example, it can promote the cancer survival because it can produce the EGFR signaling activation and also TGF-beta activation. And also ADAM9 can promote the cancer metastasis. And also it can avoid the immune attack by avoid the natural killer cell recognition because it can collect some ligand on the cancer cell to avoid the NK recognition. So ADNI is a major target for cancer treatment. So let me briefly introduce what we finding briefly. Basically, we found the ADNI dependent KRAS feed-forward loop, and that can to promote the tumor growth. And when we knock down the ADNI, it can promote KRAS degradation. That is because knock down ADNI can increase the plasminogen activator inhibitor. This PI1 has a novel function as a selective autophagy receptor. So this PI1 can direct bind with the KRAS and also bind to the autophagy component LZ3. So it will bring the KRAS into the lysosome degradation. And our developed small compound ADNI inhibitor can promote the KRAS degradation. And also we found it can enhance the chemotherapy gemcitabine treatment. And we proved it a therapeutic effect in preclinical animal models. So next, I will provide some evidence to support our finding. So first question, does the KRAS wild type also show the RAS feed-forward loop? In TCGLD database analysis, we found that the KRAS wild type has a better survival than KRAS mutant. However, when we divide the wild type KRAS into low and high expression, we found that the high KRAS wild type has a poor outcome, just like the KRAS mutant. So, and we also find the high KRAS wild type also conduct the KRAS downstream signaling. So that means the KRAS wild type still transduced the feed-forward loops. So next, we want to identify the mutation-independent RAS activation modulator. That is related to high KRAS activity, but also related to high KRAS wild type expression. So after analysis, we found the top two candidates. One is the ADGR-G6. This is the G protein coupled receptor. The regulation is very complicated, so it's not a good target. Another is the ADAM9. So next question is ADAM9 a KRAS modulator? So when we knock out the ADAM9 in mouse tumor and then transplant into the mice directly into the pancreas, we found the ADAM9 knockout reduce the tumor growth. And then we collect the tumor samples. We found ADAM9 knockout tumor reduce the total KRAS protein and also reduce the active form KRAS. And this phenomenon was observed in the cancer cell cultured in serum starvation condition. So next question is what's the mechanism? So we struggle there for long time because current KRAS degradation mechanism cannot fully explain our finding. So we try to find what's the RAS interaction protein, particularly in the ADAM9 knockout cells. So we're using the pan-RAS antibody to pull down the RAS interaction proteins and identify them by mass spectrum. And we found in the ADAM9 knockout cells, we found the SERPIN-E1 is the gene name, the protein name is PI1. We found that PI1 is enriched in ADAM9 knockout cell interact with the RAS. So next question, can PI1 increase the RAS protein degradation? So we found in the ADAM9 knockout cell, we found the PI1 level is increased and the low KRAS proteins. When we further knock down the PI1, we found that it rescue the KRAS protein expression. And in contrast, when we overstretching PI1, we found that it reduced the KRAS and pan-RAS. And this phenomena can be rescued when we treat with the lysosome inhibitor. but not treat with the proteasome inhibitor. That means the 8 and 9 knockout mediate, that means the PI1 mediate KRAS degradation is through the lysosome degradation pathway. So how can PI1 lead to the KRAS degradation in lysosome? Because autophagy is one type of protein degradation. So our hypothesis is it's possible PI1 is a selected autophagy receptor, so it can bind with the LC3 and then bring the cargo to go to the lysosome degradation. If so, we should detect the LC3 interaction regions. We call the LIR motif on the PI1. So indeed, we found the PI1 has the revolution conserved the LIR-like motif DWV located on the alpha helix. And we're also using some virtual docking to find the PI1 and the LC3 can interact at the alpha helix region. So to prove PI1 contain the LIR motif, we mutate the LIR motif, and we also proved only the wild type PI1 can interact with LC3, but not the mutant. So next question, how can 8 and 9 reduce the PI1 expression? In our previous study in the esophageal squamous cell carcinoma, we found the membrane 8 and 9 under hypoxic condition will translocate into the nucleus and occupy on the PI1's promoter by stop the transcription factor effect to reduce the PI1 expression. So is this also true in pancreatic cancer? In fact, in the clinical cancer patient's tissue, we found 8 and 9 can be stained in the nucleus and also with associated with the high KRAS expression, but the low PI1 expression. And we also proved the 8 and 9 can occupy on the SERPIN-E1 promoter. So next, we try to develop a small compound to block the 8 and 9 protease activity by using some virtual docking and do some small compound screening test. And then we find the best efficacy compound. And we found that this compound treatment can reduce the KRAS proteins and activity in dose-dependent manner in multiple KRAS mutant type like G12D wild type and G12V, G12C. And the binding affinity about our 8 and 9 inhibitor is around the 6 micromolar. And this inhibitor is specific silence the 8 and 9 protease activity, but not the 8 and family member. And we also test whether this compound can be used in clinical specimen. So we collect the 4 individual patients' tumors and they treat with the drug. And we found the combined 8 and 9 inhibitor with the gemcitabine can successfully reduce the KRAS proteins, but increase the PI1 proteins. And we also we also developed the patient-derived cancer cell, and we found this cancer cell is a KRAS wild type and with a high KRAS activity. And we found the 8 and 9 inhibitor has a beta treatment than the gemcitabine, and combine both of the inhibitor with the gemcitabine has the best treatment effect. 70% tumor disappear and induce a strong cell apoptosis. And the natural cancers have the news to report our contribution. This is the right now some strategy to target KRAS, and our 8 and 9 inhibitor can promote KRAS degradation regardless the KRAS mutant status, over benefit just like the Pan-RAS inhibitor. But there's also two questions. Is this only specific to pancreatic cancer? Can it apply to other cancer type? Or can this inhibitor also activate the cancer immunity? We are now working on to address these two questions. And the clinical KRAS inhibitor usually develop the resistance is because the EGFR activation. And when we compare with the current drug, we found the G12C inhibitor in low dose treatment can enhance the EGFR phosphorylation. But our inhibitor not only reduce the KRAS, but also reduce the EGFR phosphorylation. That means our inhibitor may not have less potential to develop the drug resistance. And also, our inhibitor found can suppress the cancer metastasis in other cancer types, like the lung cancer metastasis and the triple-negative breast cancer relapse model. We found our inhibitor can prolong the mice survival, just like the human disease-free over 20 years. So finally, I will thank the grant agent and my team members' hard work, and the special thanks to the Dr. Minji Hong's guidance. And I will stop here, and thank you for your listening, and I'm glad to answer your question. Thank you.
Okay, so that's interesting that you identify this lysosome actually through the Pi1 guide the KRAS to this LC3 mediate degradation mechanism. So I have a kind of question. So because your inhibitor kind of block the protease activity of the ADAM9, right? And so that you actually drive the Pi1 expression so it can degrade the KRAS, right? So, however, when you actually have your model set that your ADAM9 bind to the, the Pi1 promoter, right, to block the Pi1 expression. So the question is, how could the drug, which actually, if that is affect the activity, right, and this activity probably perhaps affect this membrane form of ADAM9. So how could this inhibitor actually activate Pi1 to, looks it actually hard to imagine that activity of protease actually involved in the gene transcription. So I think you probably have some answer to explain. And secondly is that TGF-beta actually is an inducer of the Pi1. And Pi1 previously shown is a senescence marker, right? And the question is, does the TGF-beta could actually drive the KRAS degradation?
Okay, thanks to two important questions. So the first question is how, because in some conditions, the membrane ADAM9 will translocate into the nucleus, right? So how can our ADAM9 inhibitor have the function to block the ADAM9 function? Yes. I didn't test whether my ADAM9 inhibitor our ADAM9 inhibitor go into the nucleus. We didn't check that. But maybe because our compound is small compound, so it could be pass into the cell and maybe go into the nucleus to suppress the ADAM9 function. But we didn't check that before. So we will check that. And the second question is because TGF-beta is inducer to produce the Pi1. So most of the scientists think the Pi1 has the two functions. One is to promote cancer, the other is anti-cancer. But most of the people think it is to promote cancer. But in our system, we found under 8 and 9 knockdown system induced the PI1 expression is play the anti-cancer function, especially go into as the selective autophagy receptor. So we think maybe PI1 under 8 and 9 knockout condition have some modification that are different from the regular PI1. So that's my explanation, but we still don't know the answer yet because we will try to find any modification of the PI1. Yeah, thank you.
Thank you. That's a very provocative answer. Thank you. Dr.
Yeah,
Wu?
I have one question. I think very similar to Dr. Lin's question. I think first of all, this G protein, this one is occurring in every cell type, every location. And did you have a chance to look at the so-called so-called toxicity or with other normal tissue or cell, or maybe also you should do some proteomic study after this treatment between the cancer cell and the so-called normal cell. Do they have any change of proteomics effect? Because that would be consequence effect, whether this drug could be used appropriately.
That's a great question. Yes, we have tested the safety about the 8 and 9 inhibitor, and our inhibitor is subcutaneous injection daily for 2 months, and we didn't see any side effect for the mice system. And we check it didn't change the immune cell population. We test the CBC and test the kidney toxicity and the liver toxicity. We didn't see any toxicity about the mice. So I think our drug is quite safe in animal models. And about the proteomics to see any potential toxicity, we didn't do that yet, but we will do that. Thank you.
Great, thank you. Last question, Dr. Kang. Xinjian.
So sorry about that. So when this ADAM9 binds to the promoter, it inhibits, it becomes a suppressor, right? Okay.
Yes.
And how does it work? Is enzyme activity required?
And yes, we check our experiment and we found it's not the enzyme activity is important because under hypoxia condition, 8 and 9 will translocate into the nucleus. But if we mutant the protease active domain, it will reduce the 8 and 9 go into the nucleus. So we think the enzyme activity is important for nuclear translocation. Yes, and when he goes into the nucleus to occupy on the SERPIN1 promoter, it will interact with the transcription factor, and then to interrupt the transcription factor's activity. So it will just like the replace the transcription function. Yeah, that's how it works.
Great. Thank you very much, Dr. Sher, for the interest of time. Just great, all the great questions and the next discussion. Thank you.